Okay, for our next section, we are going to start a chapter on how cells actually release energy. In other words, how they turn molecules like glucose into molecules of ATP, which, as we've talked about before, is the main energy molecule that our cells use to do just about everything. So, to kind of paraphrase this chapter, I talk about it as being the cellular respiration chapter. Now, again, we're not talking about respiration as far as inhaling and exhaling. We're talking about respiration that happens at the cellular level inside the mitochondria and to some degree outside the cytoplasm as well to actually manufacture ATP. So it looks like a long chapter. This is probably one of the most challenging chapters in the entire course, but if we take it in small steps and break it down, it's actually not that bad. If you take the time to understand what's going on at the level that we ask, it's really not that daunting of a challenge. So the first couple of chapters are introductory, or chapters, first couple of bullet points are introductory. We're going to take a look at the overall idea of what ATP is. We're going to go back and review what mitochondria look like, and then we're going to start looking at the steps to do this, which include glycolysis, aerobic respiration, which includes the oxidation of pyruvate, the Krebs cycle, the electron transport chain, and chemiosmosis. We'll take a look at the overall yield of ATP from one molecule of glucose, and then take a look at some other molecules which can also be used at times to make ATP if glucose is not available. And then finally, the last section of the chapter deals with fermentation, which is a way to, for lack of a better term, I guess, keep glycolysis going even though oxygen might not be available, because sometimes our bodies don't have enough oxygen to continue aerobic cellular respiration. Anaerobic respiration can continue, but only if we have a way to maintain glycolysis. And so that's the last section of the chapter is to look at fermentation. All right, so to look at the overall picture of what photosynthesis looks like, right? So if we start off and review the equation for photosynthesis, it's going to look like 6 molecules of CO2, 6 molecules of water, in the presence of sunlight are going to yield 1 molecule of glucose and 6 molecules of O2 gas, right? So if we take a look at that, that's kind of the breakdown. So actually what I'll do is I'll switch gears here for a second, and we'll make a little Word document here to take a look at what we're dealing with photosynthesis versus cellular respiration, because they are very much related to each other. In fact, they are more or less the reverse of each other. So we'll take a look at that. So for photosynthesis, we are going to have 6 molecules of CO2, and we're going to have 6 molecules of H2O, and that's going to give 6 molecules of O2 gas and one molecule of C6H12O6. And so we go through here and make some subscripts that should help us make some sense of this. It looks more like we would write it actually in class. Once we have some subscripts here, that should help make it look a little more chemically appropriate, I guess you could say. So we'll do that. Got our subscripts going here, and then in just a second, once we're done doing this, we're going to look at the equation for aerobic cellular respiration. Okay, so that's the idea here that we're looking at with photosynthesis. And so if we go ahead then and shift gears and look at cellular respiration, we will, let's see if we do it this way, aerobic cellular respiration, aerobic cellular respiration rather, is going to start off with 6 molecules of O2. Actually, I'll do it this way to save us a bit of time, hopefully. 6 molecules of O2. A little bit awkward, but we'll get it done here. And then we're going to have also some glucose. So we'll take our glucose formula here, add that to the equation, and then that's going to give us, man, we have this down by now. That's going to give us basically this whole side of the equation over here. Okay, so it's going to give us 6 molecules of CO2 and 6 molecules of water. And so that is the relationship between photosynthesis, which we've already covered, and aerobic cellular respiration. And if you'll notice, they are basically the reverse of each other, right? So we take this one, start off with CO2 and water, that gives oxygen and glucose. This one, we start off with oxygen and glucose, that gives us CO2 and water. So that's just a really long and kind of awkward way of saying what's going on in this slide right here, and that's that photosynthesis and glucose are the opposites of each other. Right? So glucose, which is made during photosynthesis by autotrophic organisms, is a really important medium-term energy storage molecule. Most cells, most of the time, at least if they have oxygen available, are going to default to breaking down glucose to provide ATP. And if glucose is not available, then we'll start looking at some other things like fats and proteins and maybe even nucleotides from now and again to actually make ATP. All right, so that's where we talk about other organic molecules, things like proteins and bigger carbs and even fats and nucleotides being used to broken down into glucose and eventually ATP. All right, so we looked at the equation. We're going to move in then and start talking about what we're actually going to do to glucose. Now, glucose, I like to imagine as being an energy storage molecule, kind of like a battery, right? So we're going to take sunlight, we're going to charge it up, and we're going to make a molecule of glucose, which represents stored up sunlight. So if we take our glucose, which represents stored up sunlight, and very carefully, very gradually break it down, we can then release the energy that was stored up in the glucose originally. Right, so glucose represents stored sunlight. If we break down glucose, we get some of the energy back out again. As we've seen in previous chapters, energy can be converted from one form to another one. Right, so what's happening in the breakdown of glucose is that we're going to take electrons out of glucose, and these electrons are high energy electrons. And so one thing you want to think to yourself is, okay, well, why are these electrons high energy? And the reason for that is because the energy was put there by the sun. We take these high energy electrons, we extract them from glucose, we handle them very carefully, and that's going to be used to make ATP. Now, if we think back to one of our earlier chapters, all the way back to chapter 2, we talked about electronegativity, and we talked about affinity of different types of substances for electrons. Some things like electrons more than other ones. So if we set up almost like an assembly line or a chain of different types of molecules, each one that likes electrons a little bit more than the one that came before it, we can actually pass those electrons along like a relay race from one molecule to the next, to the next, to the next. And every time we move the electrons from one acceptor to another acceptor, they're going to give up a little portion of their energy. Okay, so what we're talking about here is redox, or breaking down molecules and passing electrons from one molecule to the next. Eventually, the very last one in line, at least in terms of dealing with aerobic cellular respiration, is oxygen. And oxygen is going to be the one that likes electrons more than anybody else. It is going to be what we call the final electron acceptor. And that, again, is the case only when we're dealing with aerobic cellular respiration. Right, so our overall equation for aerobic cellular respiration is this. We're going to take glucose, we're going to take oxygen, and we're going to create CO2, water vapor, and energy. All right, so what does this mean in terms of everyday lives? Glucose comes from the food that we eat, O2 comes from inhaling air, CO2 goes out when we exhale air, water vapor goes out when we exhale air, and energy is used to power our cells. Right, if we look at it energetically, what it breaks down to is this. Under ideal conditions for actually a prokaryotic cell, you'd be looking at 36 ADPs and 36 phosphates, giving us 36 molecules of ATP. And that's really the main molecule that we're interested in here is the ATP, which is the energy up there in that equation. Right, so looking at cellular respiration now broadly, we're going to look at three main steps. Right, glycolysis, which literally means the breakdown of glucose, is an anaerobic process that occurs in the cytoplasm of the cell. It actually does not require the mitochondria and does not require oxygen. Glycolysis is the only anaerobic process we're going to see in this entire sequence of events. The point of glycolysis is to create actually three things. It's going to give us some ATP, as we see over here in this picture, 2 ATPs. It's going to give us 2 molecules of something called NADH, which is an electron carrier or an electron shuttle. And it's going to give us 2 molecules of something called pyruvate, which is a 3-carbon sugar. So starting off with a 6-carbon sugar, we create 2 3-carbon sugars, 2 ATPs, and 2 molecules of NADH. And again, this is glycolysis, an anaerobic process occurring out in the cytoplasm of the cell. The next really big step is the Krebs cycle, and this actually does occur inside the mitochondria, and this one is an aerobic process. So the Krebs cycle is going to generate a whole bunch of stuff for us. It's going to create 4 molecules of CO2, 2 molecules of ATP, a whole bunch of electron carriers, 6 NADHs, 2 FADH2s, and those molecules are going to follow into the electron transport chain. The last really big step of aerobic cellular respiration is the electron transport chain, and that's where we're going to take all of these electron carriers that we made over here, and there's going to be about 10 NADHs and 2 FADH2s. All of these are reduced forms of electron carriers. We're going to funnel all of these into the electron transport chain, and from them make a grand total of about 34 ATPs, most of our water vapor, and this again is where oxygen is going to be used as well. So that's a really quick, 1 minute or less description of what happens with aerobic cellular respiration. There's one other step I didn't mention. It's a little bit more of a minor step, but a very important one nonetheless, and we will get to that one a bit later on, where we take the pyruvate, move it into the mitochondria, and oxidize it. That's the 4th step, which I didn't mention on this slide because the slide doesn't really emphasize it too much. Okay, so most of the ATP we're producing in this process does occur in the electron transport chain, which I'll refer to typically as the ETC. The rest of the ATP that's made, 4 in the Krebs cycle, 4 in glycolysis, is a relatively minor component. So most of the ATP does come out during the electron transport chain. All right, so we need to go back and take a look at the mitochondria, review the structure of these because it's been several chapters since we've seen those. We're going to see what they look like. All right, so mitochondria, as we know, are membrane-bound organelles that only show up in eukaryotic cells. They do not show up in prokaryotic cells, and they have a fairly complex little structure. Right, so there is an outer mitochondrial membrane, which we'll see here. There is an inner mitochondrial membrane, which is the brighter yellow line in this picture, and then all the way on the inside of that is something called the matrix. Right, and so we also have in between the two membranes what's called an intermembrane space. So going from the outside in, we have an outer mitochondrial membrane, an intermembrane space, an inner mitochondrial membrane, and then all the way in the center we have something called a matrix. From the inside out, matrix, inner membrane, intermembrane space, outer membrane. And each one of these parts of the mitochondria is going to be responsible for doing a different part of aerobic cellular respiration. So they all have their own role to play when it comes to manufacturing ATP from glucose in the presence of oxygen. Okay, so we're going to take a look at glycolysis next. And glycolysis is the first big step of the breakdown of glucose to manufacture ATP. Glycolysis, like I mentioned before, is an anaerobic process, and it does occur out in the cytoplasm of the cell. Right? So this first step up here, glycolysis, breaking down glucose. We're going to split glucose in half. We're going to make 2 3-carbon molecules of something called pyruvate, and also some ATP and NADH. Right? So that's the first step. Now, glycolysis is actually a fairly complicated series of biochemical reactions, about 10 different steps. And in this course, that is not what we need to look at. We do not need to look at that degree of depth, so we're not going to. We're going to call glycolysis just one thing and imagine that we're feeding glucose in, getting pyruvate out, and the process also manufacturing some NADH and some ATP. Right? So that's glycolysis for our purposes. This is cytoplasm, again, and this is also anaerobic. The overall production of ATP from glycolysis is only a net increase of 2. And the reason why I say a net increase of 2 is that it takes 2 ATPs to do glycolysis. It requires 2 ATPs. Glycolysis actually gives us 4, so the net result is plus 2 in the ATP column for the overall production of energy. The way these ATPs are made is through a process of what's called substrate level phosphorylation. The reason I mention that is that even though the ATP that comes out is the same, it's a different mechanism to make ATP than what happens later on with the more aerobic steps in cellular respiration. So it's a different means to produce ATP. We're not going to worry too much about what that means as far as how it works, but just know that it is a different way of manufacturing ATP than we see later on with these ATPs made down here, for example, from the Krebs cycle and from the electron transport chain. Right? So we're starting with glucose, doing glycolysis, making ATP, making NADH, and making pyruvate. Right? So another thing to think about here is that when we're making the NADHs, what does that actually mean? Right? So what we're doing here is we're taking a molecule of something called NAD+, and so we'll switch back to our Word document here for a second, and we'll see what that looks like. We're going to say the reduction of NAD+, and that plus sign should be superscripted, so we'll do that. Right? So we'll start off with this equation here, and we're going to go down here. We're going to start off with NAD+, and so we'll do that, and we're going to add to that a molecule of H+, so one hydrogen ion, which we've seen before. So we've got that, and we're going to also add to the mix 2 electrons, and that's going to give us 1 molecule of NADH. And this reaction actually is reversible, so we'll go ahead and do that as well. Right? So this is our overall reaction for the reduction and oxidation of NADH. Okay, so if we take a look at which direction is which, the left to right arrow up here, so we'll label it this way. If we go left to right, that's going to be reduction, because we're adding electrons onto something, that's a reduction. And if we go from right to left, that's going to be an oxidation. So remember the definitions of oxidation and reduction. Reduction is when you add electrons to something, so if we add 2 electrons to NAD+ and H+, that is a reduction, and that gives us our NADH. If we oxidize something, we're taking electrons away from it. So if we take electrons away from NADH, release those, release an H+, release NAD+, that right to left direction is oxidation. Right? So we've seen those terms before, but I just wanted to remind you guys real quick what that means. All right, so in terms of filling in the blank on this slide, the two NADHs are produced by reducing, or the reduction of, NAD+. By reducing NAD+, by adding electrons to it, we create NADH. And NADH, like I've talked about before, is like a taxi cab or a shuttle or a carrier of electrons where they can go from point A to point B. And so you might wonder, well, why do we want to move electrons from point A to point B? Well, electrons carry with them energy. And if we want to move their energy, we also have to carry the electrons themselves. And so that's where these electron carriers come in. The NADH, and later on something called FADH2, are the electron carriers that get the electrons and their energy where they need to go. All right, so this picture shows us sort of the 10 steps involved in glycolysis, going from initially glucose, eventually to pyruvate. We see that it does cost 2 ATPs, so 2 ATPs are spent here, and we do get 4 ATPs out in the end. So like I said, it's a net increase of 2 ATPs. We also produce 2 NADHs, and we eventually end up with our 2 pyruvates as well. Right, so we're not going to worry too much beyond that about the specifics of different molecules involved as far as the steps in between, the middleman steps and so forth. But the end result of glycolysis, and again, I think what we'll do here is I'll switch back to our Word document. The result of glycolysis, if we kind of continue this on and make another bullet point here, is that we're going to get a grand total of 2, I want to emphasize net ATPs. So we spend 2 and we receive 4 ATPs out of the process. We get 2 molecules of NADH, and we get 2 molecules of pyruvate, which is, again, a 3-carbon sugar. So we start off with a 6-carbon glucose, and we end up with 2 3-carbon pyruvate molecules. Notice we actually haven't lost any carbons yet. We started with 6. We still have 6. So we really haven't done too much with those yet. We haven't made any CO2 yet, for example. So that's a fairly important point as well, is glycolysis doesn't generate any CO2, and we don't lose any carbons from the process either. All right, so one thing I want to point out here too is once we have our pyruvates, we do glycolysis with glucose to give us our ATPs, our NADHs. We get our pyruvate, but now we have a decision to make. And so the decision to make is based on whether or not we have oxygen available. Once we have pyruvate, if our cells are in anaerobic conditions, then we go into fermentation, and depending on what kind of organism we're looking at, you either make ethanol, which is alcohol, or carbon dioxide. Or if you're an organism more like we are, you go through fermentation and you get lactic acid, which is a very different kind of thing altogether. We'll talk about that later on and the different types of implications and ramifications that has too. If you do have oxygen available with pyruvate, then you go into aerobic cellular respiration, you go ahead and do the oxidation of pyruvate, you do the Krebs cycle, you do the electron transport chain, and you manufacture a whole bunch of ATPs. So the decision here on what to do with pyruvate is based on whether or not oxygen is available. Fermentation by itself actually doesn't give us any ATPs, but cellular respiration gives us quite a few. So this is the preference for the cells most of the time, but as long as they have oxygen available. All right, so we're going to go ahead and stop this section here, and I will start up with another section here in just a second.